Okay, good deal.
We're starting soon.
I think we're good.
Well, I'm saying we could just have it like this, but you could just stream straight into BioNanomics. Say that. You could just stream straight from your laptop to BioNanomics instead of me doing this.
Oh. Why don't I just record it? We don't need to live stream. Let's just record.
Sure. Well, it
Yeah.
wouldn't be open my messages. I'm gonna send you we can start.
Oh. I'm giving this.
I sent it in the apples tracker on accident, but there you go.
This one. You want me to copy that?
Copy. So go to OBS.
Welcome, I'm Kate Watley. We're so happy you're here. Thank you. This is great for our faculty and students. And I'm going to give you a business card and let you get set up for your event.
Excellent. Awesome.
Thank
I'll
you.
stick around afterwards. So.
All right, we're good.
We're good?
Yep.
All right.
You keep an eye on it. Make sure it's good.
Cool.
I just sat down. Cameras on.
Testing.
You can just hold that.
Just hold it one.
Yep. And then
Testing.
if you click it, that'll mute it.
Okay.
And you can hold it, when the light goes out, then it'll turn on.
Gotcha. But just tap it and it mutes it.
Yep.
Okay, good. There's an open one.
Yeah, there's a seat over here.
You want the mic? What's that? All right.
They gave me a script.
They gave you a script. Don't go off a script. That's good. I'm not going to be on script anyway.
All right. Check 1, 2, check.
Oh, you got to tap the button.
All right. Looks like we have a packed house. We actually are having to turn people away because of fire code. So that's
Oh no.
a good sign. That's a good sign. Thank you all for coming out today. For those of you who don't know me, my name is Dr. Mike Mitchell. I'm new here, so you might not know me yet. But it is my absolute pleasure and honor to welcome Doug Horner to us today. He's joining us from right down the road, down at the beautiful Electric Work campus, where Doug started a little company how many years ago?
Oh, 30 years ago, yeah.
30 years ago, called MIE. That stands for Medical Informatics Engineering. And I've had the privilege of knowing Doug now for the last few years, and I can tell you, you don't often find people that work as hard and play as hard as he does. But he plays hard on geeky, nerdy things, and it's after my own heart. It's just, he has a huge passion for teaching the next generation of scientists that I've only encountered in a handful of people in my entire life. And so it's really neat to see somebody driving as hard as he is to help educate you. Other ways that Doug's involved in this education is he has a not-for-profit called BioNanomics, And this company is there to give you access to toys, so scientific equipment, toys that normally you would not have access to in Fort Wayne. This BioNanomics company makes them available to you. And so seriously, you can show up and he'll put you on something if you have an interest in science. Not only that, currently you sponsor 7.
Yeah, 7.
So 7 FIRST robotics teams. So these are the robotics high school teams that compete, and there's even some middle school teams coming on board. And I got to see the robots over the weekend. And they're big and they're fast and they're borderline scary.
Dangerous, yeah.
They're actually physically dangerous. But it teaches kids programming, robotics, problem solving, team skills. Our local team, Snyder, went to Worlds last year. And we're hoping.
And Huntington did too, yeah.
Huntington, I guess, I learned this, is a perennial powerhouse when it comes to robotics competitions. I can tell you, Doug, you have a deep passion for science, putting pieces together, and how do we marry biology and technology to make everyone's world a better place. And I really appreciate you coming out today. So if we could give a nice tech welcome to Doug Warner, I appreciate it.
Thank you. All right, thank you very much for coming and listening to me dribble on about tech. So just a quick show of hands, how many people here are in the life sciences biology side of the campus? Okay, great. How many people are on the tech side? Computer science, engineering. Oh, you're in the minority today. And I'm curious, can anyone holler out what other disciplines are out there? Did I miss anybody? No? Okay, good. Well, I like to give this presentation, depending on who the audience is, and I actually gave a good tech one on the tech side. There's a recording actually I'll show here. These are past presentations that I've given, and so my background is in software development and health informatics. So that's what medical informatics engineering does as electronic medical records. We manage about 250 million medical records in 70 countries. We manage, in 12 different languages, and that's what my company does. But back in 2001, when they sequenced the human genome, I started looking at the human genome as code, right? And it is a quaternary code that's no different than the code that runs the computer. And so that's when I started to really get interested in virology and genomics and epigenomics and things of that nature. And so most of this is sort of self-taught. But one of my heroes here is Richard Feynman, and this is a picture here in 1947 of the first transistor. And in 1959, he said there's plenty of room at the bottom that we should start developing small motors. In fact, he offered a $1,000 prize for someone who would build a motor that was 1/64th inch size, and I think within 3 years he had to pay that $1,000. So, but he had basically calculated that there's plenty of room at the nano scale to build robots and machines. And what's been amazing to me is that as we've gotten better at probing the nano scale, we found that the robots were already there. So the purpose of my talk here today is to inspire everyone to think differently about biology and synthetic biology in particular. I want to show, I'll do this. We'll show you the lab that we have at Electric Works. And I have, this is just one corner of the lab here. And we have a couple of these segues here because it's a big campus with lots of space going around. But there are 3D printers. We have a flight simulator and a virtual reality system, including these things that can push back. That's like a virtual surgery system, so we can actually manipulate cells under the microscope and feel the tissue pushing back at us. And I'm going to fast forward over to the lab part. Actually, we'll go back here. I think I have pictures of the electrons. Yeah, here we go. So this is the University of Michigan's electron scanning microscope here. And we actually relocated it down here. We got a deal on it because they were getting rid of it. And then we built this room out. So this was actually taken last week. and that equipment's being moved in. And then this here is the actual confocal, well actually we're going to show a atomic force microscope here. So it's floating on a set of bungees, because even walking around it, it would cause degradation in the imaging signal. And I'll fast forward here. This is actually the confocal microscope here. So there's a laser stack here. This is an optical floating table. There's actually a live imaging, so you can actually image things live, and both an infusion system there to actually inject drugs at different points, as well as a system for controlling the CO2 and O2 levels and things of that nature. So, and then I'm not going to show you guys this. We'll show you that we'll do a quick meet the microscope.
All right, I've got some cheek cells on the microscope. I just cut the nucleus out of the center of a cell. Watch me do it. So I'm going to find another cell using the hand tool here. I'm just going to pan over to another cell. Now I'm going to use the drawing tool here to draw a circle around the cell.
So literally we have a system where you can draw right on the cells, and that's a laser system here. An arm and a laser that's on the back of there. And then up here, this will stop blinking when the laser's warmed up.
So with that circle, that button will cut that out. So now I'm going to go hit the cut selection. And now we'll see that this is going to warm up.
And you'll see the laser's actually now cutting into the tissue, and it's getting just that nucleus there.
So we're going to hit it again, and it'll pop right out
And
there.
I decided to hit it twice there, because if it's not, if it's connected at all, it won't come out.
And it will stamp up and pick
So that little stamp has a bit of stickiness on it, and it attaches to the scope.
Thing here, so this just magnetically attaches. It's hovering above that. And now I'm going to hit this button up here in the corner.
I can't see it. There's a little button behind the little thing
You'll
here.
see it just stamped down on it.
And the stickiness stamps down on the tissue.
Button here. And now
And
there's
then
a
it pulls
hole
that
where there once was a nucleus.
individual
It's
part of
on
the
that
tissue out.
cap.
So what's really cool is, in a second
now
I'll show the
you come
cap.
over here,
You just pull the
take
cap
this
off,
cap
and then
off,
that individual cell with that nucleus
on that lid.
is on that
It's
lid,
small
and
to
then we can
see,
send it off to
but
be
that nucleus
amplified and
is now on
sequenced.
there. I could
We have a
close the
nanopore
cap up,
system coming in, so we
and
could
now
actually
adjust that individual cell
read the
nucleus
individual
inside
DNA
that
and see
tube.
which sequences are
All
turned
right, I've
on,
got
which
some cheek
sequences are turned
cells
off.
on the microscope. I just
Okay, so that's a little bit about the lab there. And anybody, by the way, if you're interested in using the equipment, it's a not-for-profit charity. We just ask that you open source your findings and things of that nature. But you're welcome to propose experiments and schedule time on the equipment and run and just publish the data. Okay, so just a little bit about nanotech. Everybody, familiar there with the. This is a man-made thing here that clearly looks like an engine, and I always like to show a little bit of clock ASMR. So here's a guy, working really on a watch, putting together these gears, and literally this is an ASMR. I think they call it an ASMR thing. So for those that are out there that like ASMR, you can watch them put together clocks and stuff on the internet, right? But what's fascinating is that if you do an electron scanning micrograph that's a bacterial flagellum motor. So that's actually what's driving the bacteria, and it's essentially a motor. What I'm going to do is I'll show you guys a little bit.
15 microns long and spins on the order. Fle-G, along with Fle-M and Fle-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. Mote A and Mote B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but Mote B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The assembly of the hook proteins.
Is also show, I'm going to fast forward. You guys can all come back and watch these videos.
After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and...
But what I'm going to do is talk over it here. So what you're seeing are proteins essentially being fed through the center channel of the flagella, and actually building the flagella out. And essentially all these are basically like little Lego bricks, right, that are proteins that are the structure and the how they're formed are essentially programmed in the DNA. And then there are while loops within the DNA that tell you how many of these need to be produced, because bacteria that are flowing through very viscous fluid need a different size flagella than something that's from something that's very liquid, right, easy to flow through. So there's literally programming that you can watch in this video here. I won't play.
This is a nano-sized biological.
Literally, you can go in and look at and see how different genes interact with each other, and how this is programmed to actually for the speed and for the length of the flagella is all. There's a really good video here that takes you through the math. Sorry for that. There's actually math equations here. I try to limit the number of times I bring the math out. But what's fascinating is that you can computationally define these things and then simulate what the bacteria will do. So there's some good videos. Hopefully, we'll give a link out to everybody so you can come into these slides and play these videos in full. I'm going to skip this slide. This is for the geeks out there that might not know what DNA is. Hopefully everybody here knows DNA and how DNA is expressed from DNA into RNA and then from RNA into protein. So we won't cover that here today, but I love some of these videos. Anytime I can get the Amoeba Sisters videos or Stated Clearly. How many people know Stated Clearly or the Amoeba Sisters? Oh, good. Everybody's, yeah. Those are both excellent channels to follow. I love everything they do. How many people know the acapella science? Any show of hands? No? Okay, I'm sorry. We have to play just a little bit of this.
A dot isn't the best way to try to sum up how electrons come and go. They are the states of a matter field that follows an equation, the Dirac probe. The Schrödinger part of the whole equation will just lead in sub C when it expands. Now get that cool thumb and add it in with the proton and watch them start to dance. It's hydrogen, it's like, oh proton, I feel your tug. Central potential, dip down, pulling on me. But I'm not falling in deep. No, that would break uncertainty. Say, oh, electrons move too much. Slow down your pace and put that orbit on me. Come on now, follow my lead. Come on now, follow my lead. Orbitals take the shape they do, the stable states of the quantum rules. And when one approaches two, they combine and they're bonding. Thus hydrogen as a rule is found in nature as a shoe. Energy configuring a molecule, diatomically bonding. Diatomically bonding. Diatomically bonding. Diatomically bonding. Energy configuring a molecule. When orbitals take the shape they do, have spin, no give a lift on aromatically bonding. Aromatically bonding. Look at benzene in a ring, they hold it true. When orbitals take the shape they do, come bond with me, baby, come bond. 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 Polymers take the shape they do, combining base level residues like RNAs, ACGU, like their hydrogen bonding. Peptides make a chain and group, and get a plate sheets and corkscrews with the secondary links they fold and move. They're all over your body. Come on with me, baby, come on. Come on with me, they're all over your body. Come on with me, baby, come on. Come on with me, they're all over your body. Come on with me, baby, come on. Come on with me, they're all over your body. You're a chemical machine, it's best to do. The molecules take the shape of you.
I love his content. I always have a QR code for people to go subscribe to him. He should have a couple million subscribers. I think he has a couple 100,000, but everything he does is great. But basically, he tries to. By the way, that was all a cappella. He literally did all that with his voice. My poor 4 daughters. I play this all the time. They don't even know the real words anymore. They sing these words. So, and they hate me for it. But he's making the point that we're all biochemical machines, right? There's all these little individual components that, at scale, form the cell. And this is an example of a molecular dynamics simulation that computationally is finding the binding pocket of a small molecule into a larger biomolecule. And this is all being simulated in a computer. Everything that you see here takes a few nanoseconds in time, but we wouldn't be able to see that happen. So this is a much slower version of essentially just simulating the forces between the molecules to find out where that small molecule will bind inside of the biomolecule. I'm going to skip this, but there's a really good video here that actually takes you through the different scales of these biochemical reactions and how that works. Actually, I think this is a fun one. I'm going to do, let's do this real quick. This is fascinating, right? So inside the DNA, right, this is a DNA strand here. And you can see it's coiled up on these things called nucleosomes. And these nucleosomes silence the DNA. In other words, the DNA, when it's wound up, it can't be read. So what we'll do is we're going to show a picture here. So this is actually the example of a bunch of cells sitting in tissue here. And what you're seeing is all female cells have 2 X chromosomes. A male has an X and a Y, right? In every single cell in a female, one of the X chromosomes has to be silenced and put away. Otherwise, you can have problems. If you have 2 different copies of the genes open and running, you'll actually produce too much protein, or there would be downstream consequences. So, at birth, there's a process where they randomly silence and crunch up one of the copies in every single cell, and it happens somewhat randomly. So I'll fast forward here. So there's an example of an active one that's open, and then the closed up one is wound up really tight. And what I'm going to do is show. This is pretty cool here.
Each X chromosome at the molecular level, which is at approximately embryo development. The decision of which chromosome.
So this happens early in the process, in the embryo, where the maternal, some cells have the maternal one open, and some cells have the paternal one. And so if you color them pink for the maternal and then green for the paternal, you could actually see on your skin, and in fact all throughout your body, which one comes from mom and which one comes from dad.
This is an example of epigenetic inheritance. The same pattern is maintained into adult.
And so this is an example of showing the skin of the different kinds of genetic inheritance that happens even within the same tissue. You have some tissue from mom and some tissue from dad. And part of this was actually discovered in calico cats. I don't know if everybody's familiar, but the calico cats, female calico cats, have 2 different colors of fur, but only the females do this. Males don't have 2 different copies because they only have one X inherited from mom and a Y from dad. So it's an example here of showing, that small little just silencing those genes changes, a big change in the phenotype, right, of what you actually see. So that's the idea that not just genetic changes can occur, but you can actually just silence or activate different parts of the gene and have a completely different output. And by the way, has everyone seen DNA copy itself? Let me just do a few minutes. I'll just talk a little bit about it, right? So every time a cell...
Using computer animation based on molecular...
Every time a cell has to copy itself, it has to copy the entire genome. And these are the enzymes that are doing that. And it's basically, feeding it in about the speed of a jet engine. Because the DNA is kind of like a zipper, it has a certain direction to it. It has to be copied in a certain direction, which you can copy right off to the right there. You can see it spinning off. But on the left side, it has to be copied the other direction. So there's these complicated enzymes for moving the DNA strand out, and then you could see it in sections. It copies, on the reverse side. So, and this happens in every single one of your cells all the time. This is happening, right now in all of your cells today as they divide and make copies. And there's some really good videos here by Veritasium that actually explores that in detail. I highly recommend, if you haven't seen that, to come back and see more of that. Let me show you this. This is that. So if a DNA strand, like if you're out in the sun or you get an X-ray that hits you, this is an example of a DNA breaking. And so the DNA would float away. So there's these spikes that go out and connect the two edges of the DNA. So this, if DNA gets damaged, this is actually an animation showing you how that DNA gets repaired. And you could see that you have to resection parts of the DNA and then, literally like break off one half of the DNA molecule here. And we'll do this. I'm going to speed this up just a little bit. But there's this amazing dance and a lot of machinery that has to come in here to fix that DNA break. And now, so that basically built an assembly to read the DNA, and literally for the geeks out there, there's a mem compare. I call it a mem compare. It's something we do in computers to compare, to go pull off one of the other chromosomes and then do a comparison to find this exact same gene on another, on the other chromosome. So it's literally now found the similar gene on another chromosome, and then boom, now that all of these things have sort of zippered together, it now knows where to start copying to fix that break. It's an amazing dance, and you can see how it's actually copying in two directions there as it's fixing the break. Now, all the things you see here, all the actors that you see playing on this stage are actually just also in the DNA, right? All the enzymes that you see, all the machinery doing this work are expressed. There's code essentially in the DNA to build these actors to actually do the self-repair. So if you have a mutation that makes it so you can't fix breaks, now you'll have breaks that can't be repaired, right? So there happens to be, there's multiple copies of that in your genome, so that there's actually multiple DNA repair mechanisms, multiple copies of it, so that way if one gets broken, you have a break there, other ones can be expressed. So, and there's some really good videos here. Okay, so I just wanted to give you guys some appreciation for how complicated DNA is and what it does. Now, when DNA makes these proteins and things, let me show this. This is an example here of HIV trying to enter into a cell. So the HIV makes this, essentially it's a spring that at the top you'll see we'll go here, this is the cell that it's trying to enter. I'm going to fast forward here. At the very top is the cell, at the bottom is this big surface, which is actually the virus itself. And then it has this spring that winds up and coils up, and because it coils, it brings together the two sides and forces the cell wall with the virus particle. And that causes a fusion, which allows that thing to enter into the inner area of the cell. So you can see it pulls it apart here. And then there are all these interesting small molecules that have been developed for treating AIDS patients. That's a small molecule that just sits there inside that pocket and prevents the spring from functioning. So if the spring, it's called, that's the T20 peptide inhibitor. It just sits right there, and that spring can't coil, and so the virus can't get in, and that's why you have to take all these. And those things get broken down by your body, so you have to keep taking them all the time as long as you have virus in your system. And this is another inhibitor that was developed to prevent entry as well, and it just sits right there. The funny thing is that's what it does there, but it also goes and binds to other places in your body, and that's a side effect, right? So if you can make something that binds right there but nowhere else in your body, that's a great drug. But if it binds it into another place and then starts making you grow another arm or something, that's a bad drug. So, but understanding how these Lego pieces move together and work together is a very difficult thing to do, because there's a lot of computation that has to happen in order to figure out how all those players bind together. Okay, I'm actually going to skip this. This is kind of a fun tool that lets you actually, and we have this equipment at the office that lets you go in and play. That lets you go in and play with these biomolecules. So you could just put on on the hat and literally pull these things apart. And that equipment that I showed in the first slide, you could actually tie it onto one of these devices and feel the binding forces. You can actually like play with it and feel what that feels like to play with.
A sample scene used.
I'm going to play just a little bit of nanobot every time I get an opportunity.
Decisional dream data. If you need a calculator, how be? Logic is like A and I B. Inputs are Karayan, that's key. And you get a photon as a reading.
Plan your nanobot to be important info scanner nanobot. Reading and writing coded data that allows freedom to do as you program it. A Turing automatic full gamut nanobot.
Oh, nanobot, doesn't that look like oh, nanobot, doesn't that look like oh, nanobot, doesn't that look like oh, nanobot.
Break me down, I am a meta nanobot. The musing model of synthetic nanobots. Working in concert as a replica of a lot. For all I try to be more than it, though sentient and organic, I am a nanobot.
Oh, nanobot,
Nanobot, yeah.
oh, nanobot,
I am a nanobot.
oh, nanobot,
I am a nanobot.
So he does a lot of good. So what's really exciting is that this is understandable, that disease, that the development, et cetera, can be understood in these very fundamental, small, machinery-like terms, right? So that's the exciting part of this. Let me go back here. So just to draw an analogy here, because everybody's biology, I thought I would show just a quick.
Throughout this video, we're going to do a desktop computer dissection using 3D animation, kind of like a dissection lab in biology class, but instead, we'll journey through the inside of this computer and disassemble every piece of hardware. We'll then use a microscope and zoom in to give you a nanoscopic view of the transistors and other structures inside. To make this video, we disassembled all the hardware inside a typical desktop computer. We desoldered and removed the components from each of the printed circuit boards and took thousands of pictures. Then, using these pictures, we meticulously 3D modeled every single component from the computer case down to the tiniest resistor. As a result, here's all the hardware that we'll explore throughout this video. It kind of looks like a crime scene where someone viciously destroyed a computer and arranged all the components. But anyways, let's dive right in. We'll begin this journey with the Central Processing Unit, or CPU, which is the brain of...
So my goal here is to inspire people to come back and watch these from branch education, because they're really good and they're very well animated. Let me just play just a little bit of this just to show you how this goes together here.
Complicated. So here's a diagram laying out the different functional sections. There are dozens of rather complicated elements in this diagram, and you can look forward to a series of videos we're currently planning which will explore computer architecture and how each of these sections work. Let's zoom into a nanoscopic view of the integrated circuit so we can see individual transistors. These transistors are incredibly small, only a few nanometers wide, and in this die there are approximately 8 to 10 billion. On top of the transistors are multiple layers of metal wires, with vias rising vertically between the layers. Together, these transistors and wires create a multi-layer labyrinth or highway, resulting in a computer that can execute billions of operations every second. Let's now zoom out and look at other sections of the CPU. To the side of each core is the shared L3 memory cache and ring interconnect. On the far right is an integrated graphics processor which functions as a less powerful GPU. In the top left is the memory controller which sends data to and from the DRAM. And finally, on IC is a heatsink with a fan to dissipate heat. And on the side of the graphics card are HDMI and display ports, a PCIe interface, and then 28 streaming multiprocessors. Each streaming multiprocessor is composed of 128 cores, resulting in a total of 3,584 cores. Each core has sections for integer and floating point arithmetic, and sections for queuing in the operands and collecting the results, and is far simpler than a CPU core. Additionally, on the die is an L2 memory cache, shared among all the graphics processing clusters, a set of memory controllers that connect to...
So what's fascinating is, like, that's all at the nanoscale. We're talking about things that are basically on the order of the size of viruses, right? And you can probe these spaces, and you're built with, like, literally, like, taking a microscope and then shining light through it down onto the microscope instead of the other way around. Like, normally you take a microscope and you enlarge things. Literally, this lithography process was by taking big images and then pushing it through a microscope in order to actually etch things out on a wafer. And so, like, as we've been able to probe the nanoscale, we're able to build these very small logic machines. And what I see as the future is being able to use this architecture to basically understand how the DNA works and the interactions of the DNA. So,
to the
This is particle life. It's a very simple particle system that creates these incredibly beautiful lifelike structures. It can be explained in a few seconds, and if you know how to code, you could write your own version of it today. It's a perfect example of emergence, the phenomenon when many simple small parts make up a larger structure that suddenly shows complex behavior. I will explain how all of this works in the second half of the video, but for now, I will just leave you with a few beautiful impressions of particle life.
So I highly recommend coming back and watching these. So the next part of my talk, just for the next 10 minutes or so, I'm going to talk about emergence. Because, like, you might think, okay, here are these little machines and these simple rules. How can that explain us and life and the complexity of everything about ourselves? And the concept is emergence. Like, at any single level, like you have, for example, in physics, we can write simple equations that guide how an electron, binds to a proton, and then how molecules interact. And these are incredibly complex equations that you can't they're so difficult to solve that you wouldn't be able to do anything, because there's even in 1 mole, right? 6.02 times 10 to the 23rd molecules in 1 mole. So you have to do that many calculations in order to understand it. Yet we have things like thermodynamics, and so you can write these other equations that work for thermodynamics or for viscosity, for Bernoulli, for example. These other equations, for whatever reason, we're able to, at certain levels of emergence, write new equations that hold true most of the time, and then there's edges where that no longer holds true. So trying to understand what shortcuts we can take, right? If we try to simulate every single molecule in a cell, won't work. But what you can do is say, look, let's try to understand how the proteins interact with each other, and then write rules that that predict the outcome of that cell at that layer, and not worry about the individual chemical parts. And that will be useful in most areas. Like most things, you'll be able to solve problems you wouldn't be able to solve otherwise. But what's amazing to me is that emergence like really works well for like even explaining complex things such as life. I'm going to skip some of these slides here. There's some good stuff here about actually how the microscope, how you actually make simple rules that actually give you emergence so that the bacteria will survive. Like you randomly change things in different cells, and some of them will automatically take on properties so that they behave well under certain conditions, and it's through artificial selection and emergence. And so through artificial selection, this is actually evolution.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet.
Does anybody know what the megaplate is? Everybody seen the megaplate? Nope. All right, so we'll just go quickly here. This is the megaplate. What they do is they put in antibiotics, no antibiotic, a little bit of antibiotic, 10 times antibiotic, 100 and 1,000. And if you hit play,
the top of it, pour some thin agar that bacteria can move around in. You see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So that's a good example of life will find a way from, what movie was that? Jurassic Park, yes, life will find a way. Through artificial selection, you can amplify certain behaviors, emergent behaviors that can overcome like amazing things here. So let's do this. And by the way, that same kind of capability, this is one of the things I'm trying to study in our lab, is essentially we're building a cellular debugger. So that's actually a fruit fly that's developing there, and it gets to a certain number of cells. All the cells are whispering to each other, and they're all virtually identical. There's a concentration gradient, but all the cells are essentially virtually identical. And then at some point, when they have a certain number of cells, they all start doing their dance, and they all move to different positions, and they all differentiate and become something else. And this is some software for tracking each cell and its movement as you're forming an embryo. And so understanding how every cell is essentially identical, has the same copies of the genes, which genes are turned off, which genes are turned on, and then how every cell knows to go to a certain position and then divide and procreate and create arms and legs and eyes and liver and so forth. And there's a lot of this cell signaling that takes place, but a lot of it is not understood today. There's Nobel Prizes for quorum sensing, discovery of quorum sensing molecules and things of this nature that we understand the homeobox. There's something called the homeobox that defines how these cell patterns are laid down. And I'm going to skip some of these.
This worm is real, but it is 100% simulated. Does this prove simulation theory? And here's the...
So this is actually C. elegans, a worm here, and I'm going to skip past. This is one of the organisms that we're studying in the lab that always has 980 cells in it for a male, and then 1,000 something 1,020 or something for females. But you can see a C. elegans here, and we're able to, there's actually something called the visible worm, where they're able to map every single cell and its position. It always has the same number of cells. But which cells take up which roles is something that's being actively studied. And you can literally, there's 300 neurons in it, and you can go into the computer and then simulate all 300 neurons inside of a computer. But really understanding how that embryo turns into those 900 cells and every single cell knows what position to take is not well understood. So that's an area that we're playing with. Okay.
The molecular neuroscience group and the laser analytics group work closely.
These all started right off. Yeah, so this is an example of that C. elegans dividing, and you can see every cell dividing. There's 4 cells there, and then it moves on as it keeps dividing and dividing. And then at some point after it's got enough, there's a quorum sense that says, okay, we've got enough. Now you be the head, you be the tail, I'll be the mouth, I'll be the anus. And then it develops from there. But we want to basically listen in on that. How many people know Michael Levin? Anybody? Nobody knows Michael Levin. Oh my gosh. If you want your mind blown, you got to watch some of his videos.
Tell us what's going on here.
So this is a creature known as a planarian. They're flatworms. They're actually quite a complex creature. They have a true brain, lots of different organs, and so on. And the amazing thing about these planaria is that they are highly, highly regenerative. So if you cut it into pieces, in fact, over 200 pieces, every piece will rebuild exactly what's needed to make a perfect little worm. So think about that. This is a system where every single piece knows exactly what a correct planarian looks like and builds the right organs in the right places and then stops. That's one of the most amazing things about regeneration. So what we discovered is that if you cut it into 3 pieces and amputate the head and the tail, and you just take this middle fragment, which is what you see here. Amazingly, there is an electrical gradient head to tail that's generated that tells the piece where the heads and the tails go, and in fact, how many heads or tails you're supposed to have. So what we learned to do is to manipulate this electrical gradient. And the important thing is that we don't apply electricity. What we do instead was we turn on and off the little transistors. They're actually ion channel proteins that every cell natively uses to set up this electrical state. And so now we have ways to turn them on and off. And when you do this, one of the things you can do is you can shift that circuit to a state that says, no, build 2 heads, or in fact, build no heads. And what you're seeing here are real worms that have either 2 or no heads that result from this, because that electrical map is what the cells are using to decide what to do. And so what you're seeing here are live 2-headed worms, and having generated these, we did a completely crazy experiment. You take one of these 2-headed worms and you chop off both heads and you leave just a normal middle fragment. Now keep in mind, these animals have not been genomically edited. There's absolutely nothing different about their genomes. Their genomic sequence is completely wild type. So you amputate the heads, you've got a nice normal fragment, and then you ask in plain water, what is it going to do? And of course, the standard paradigm would say, well, if you've gotten rid of this ectopic extra tissue, the genome is not edited, so it should make a perfectly normal worm. And the amazing thing is that that is not what happens. These worms, when cut again and again, in the future, in plain water, they continue to regenerate as 2-headed. Think about this. The pattern memory to which these animals will regenerate after damage has been permanently rewritten. And in fact, we can now write it back and send them back to being 1-headed without any genomic editing. So this right here is telling you that the information structure that tells these worms how many heads they're supposed to have is not directly in the genome. It is in this additional bioelectric layer. Probably many other things are as well. And we now have the ability to rewrite it. And that, of course, is the key definition of memory. It has to be stable, long-term stable, and it has to be rewritable. And we're now beginning to crack this.
So that's the amazing thing, is it's not just the genes, there's also electrical state being kept. In fact, one of the things that blew my mind from a talk he gave a couple years ago is that if you think about it, neurons got their trick of electrically communicating by evolving, but all of our cells actually have these ion channels communicating with each other that the neurons later actually figured out how to make more efficient and quicker by myelinating the paths. So, but it turns out like all of your cells are in constant communication with each other through electrical memory, and that memories are probably stored in your cells about what they're supposed to do. Just like I showed memory, like those memory addresses in the CPU, there are memory places in your cells telling the cells what to do. And it's not just the genome, it's also literally the state of the ion channels in your tissue. So that he's got some really good talks. There's like a 3-hour talk here. I highly recommend it with going and watching it with Lex Friedman. It's a really good talk.
Tell us.
There's another talk he gave here on collective intelligence. If you want your brain blown, you can go here and talk about how intelligence exists at different levels. In fact, there's
in many ways, biology does not over.
This is an example where he talks about the lumen of a newt, and what you do is you add more genes inside the embryo, and then the cells get bigger. But the cells still know how to build the lumen. Even though the cells got bigger, you could see how like these cells all go take up a position around and build a tube essentially for urine to go through. If you make the cells bigger, they adapt and they still make it. But then here's a situation where you've actually put 5 times the amount of genetic information in, so that the cell's way too big, and the cell actually bends around and with one cell forms the lumen, forms the tube. So there's some, essentially the idea is that each cell is, regardless of the size of it, is trying to go form something in what he calls morphospace. Essentially your hand is a bunch of cells cooperating to make a hand. And if you can talk to the cells, they can form your hand, or you can regrow an arm, or you could do whatever but you just need to know how to tell them how to do what they need to do. But every cell in your body is constantly turning over, and every cell plays a specific role that it's told to do, including like cancer. One of the things he's studying is how he did this in the newt. He actually put cancerous tissue and was able to program that cancerous tissue to play nice and not become a tumor. By electrostimulating it. So he's got that's actually in this talk here where he talks about that too. So it's fascinating that we're finding other places that state is stored within these cells.
In many ways.
Okay, let me introduce. I want to switch over here and just talk. You want to take a moment? Yeah, we're gonna. I just want to talk. Oh. I think I ran the battery down. Does it? It's blinking red. Let's see, testing. We'll just have to speak. Oh, there we go. Wait, testing. There we go.
Cool.
Go ahead. I'll queue up your slides here. Sure. Introduce yourself.
Okay. Hey, I am Brianna. I am the lab manager at BioNanomics. I have been working with Doug since March. We got incorporated as the lab in July, so we're just getting our feet under us. I have worked the past 5 years in microbiology. That's my background. I got a microbiology immunology degree from PFW, and then I worked at Lutheran Hospital in the lab for almost 5 years, working on your COVID samples and stool samples and all that good fun stuff. But now I'm here with Doug since March, and we've been having a good time. Today, though, I'm going to talk about iGEM and this competition that they hold. They've been holding it since 2003, so over 20 years now. And it's something that is international. It's the International Genetically Engineered Machines, I think.
Yep.
Yep. And what you do is here. Up here really quick.
Yeah, go ahead.
What you do is you assemble a team. You've got some PIs, principal investigators, which will be teachers, and then you have advisors as well.
Here, wait. Just yell.
Yeah. I'll scream. Okay. And you gather some students around. It doesn't have to just be biology students. It's actually important that you have engineering students, you have digital design, law students, things like that. Hello? Okay, cool. Yay. so that you have a well-rounded experiment and project. So you start with figuring out your research topic, your project topic. You build your designs, you test them, and then you build like a wiki page and presentations and videos, and it goes into the competition. If you make it to the finals, they have a whole big, what's called the Grand Jamboree in Paris every year in October. And that picture right there is a picture of all of the teams and all of the people that were participating that year. So it's pretty big. And then you present your project. And there are medals and things like that you can win. And there are a bunch of different, what they call villages, basically topics that you can, like categories that you can have your projects in. There are so many different kinds. They've got some really fun ones like fashion and cosmetics. So it doesn't have to be like, just your basic antibiotics experiments. It can be something fun like that. Food, nutrition, conservation, space, software and AI. And they've been growing quite a lot. We've got lots and lots of teams started, and over just in the last 2 years, they've grown by like 2,000 participants, like over 50 teams extra, which is pretty cool. And they have like a set of deliverables that you show, and like a timeline on how to do those. It's basically you start in about February, March. The latest registration deadline is May. So if you guys want to approach me, we can talk about maybe getting a team or just learning more about it. We can do that.
Maybe we should do a team.
Yeah. And then you work over the summer for the most part. The majority of it is done over the summer, and then it has to be pretty much finalized and have your presentations ready by October, because that's when the competition is. This is the team that won first runner-up for undergrads last year. They have done a lot. So there is the biology aspect where they are developing like a lateral flow assay to detect a certain type of golden algae in their local river, which killed like 1,000 kilos of fish in just a couple of months. So it was a problem that they wanted to tackle to save their, personal, like local ecosystem. And then they had engineered and 3D printed a device so that you could take this lateral flow assay and use it in the field and get a result really quickly and easily, and it's reusable. So they engineered that as well. And then they also go around with their students and their team and raise awareness, and they've gone to multiple conferences and they've documented that whole process, and that was important as well. Let's see, and then
Skip that.
produce trash.
You got a good produce there.
Anyways, so the competition fees are pretty steep, but we would like to sponsor them as well. There are also other ways that you can sponsor your teams. You can do crowdfunding. You can go to companies like Eli Lilly, stuff like that. Propose your case. So that's where a business major would be. Marketing business would come in handy. They could help you with that, make your business case. You could do that. And then another way to get some funds back is you can actually turn your project into a startup company. iGEM is part of an international conglomerate of synthetic bio companies, and they have an event at the Grand Jamboree every year where they connect people that want to put startups in with all of these other organizations that would be able to help connect you to the people that would help fund and invest in your startup. So that's another way that you could get some of your money back on the iGEM project. But it's really fun. It looks really interesting. And even if you don't advance really far, if you don't get any awards, it would be a really fun experience, something really nice to put on your resume. You participated in an international synthetic biology competition. That sounds really cool. And just a good way to build community in your university outside of just, life sciences department. So I was here to talk about that, and if you guys are interested, shoot me an email. We'll have it up.
Go bug your teachers.
Bug your
them
teachers.
Get involved. Yeah, so if you want to do it, it's good. I think it's really good for the resume. I think it's also good because there are companies that get involved in this and sponsor. Those are places you're going to want to work. So
Sure.
like by doing this, you're going to be rubbing elbows with companies that are in the life sciences funding that kind of research. So
Be very interesting.
Cool.
That's all I got to talk about.
Well, thank you.
I know that some students
Excuse me.
some of our students obviously have a limited class, so they've been speaking out, so we want to make sure people can get to their thoughts. But obviously the team is going to hang around and chat for a little bit if anybody wants to talk to them. So thank you so much for being here.
Thank you.
I can stay and chat for a little bit, but we have Amanda.